You have very good ideas and you conducted excellent experiments. So I would like you to translate this to something which is applicable. So in your perception, what are the 2 to 3 practical things that are applicable in the daily life of our people, especially in the Philippines? And the order of answering, we start with the ladies first, and then next one will be by alphabetical. Hindi ko naman sabi kung kapugian, di ko. Kaya alphabetical. Bahala na kayo mag-estimate kung sino yung guna, di ba? Alright, yes, ma'am?
Thank you. As the minority in this group, okay. Of course, like the work that I did is targeted practically to our farmers. So the 3 main goals is to not only provide them with the direct recommendation, but ultimately to help them build their capacity as they implement this decision making in, let's say, if I have a field and they have, or like a field, agricultural field, what am I going to use to help mitigate or minimize the losses from pathogens, from plant pathogens? And among the different or the many options for use in managing these problems, which of them would be the most effective? And so with the work that I did, it's basically to target their decision making so that they would be, like smarter in terms of helping them mitigate potential losses from the plant pathogens that can build up in the agricultural crop production system.
Thank you.
Good afternoon po. So I'm pure mathematician and we get this question a lot. So saan ba ginagamit your math? This is foundational. So I think many applications would ultimately rely on our understanding of the foundations. And the work that I'm doing is trying to push the boundaries of our understanding of symmetry, which is important. Moonshine in particular is related to high energy physics and quantum gravity, but I'm not a physicist, so I don't really know if they will find applications to what I'm doing right now. But that's the hope, eventually, that it will find an application.
Thank you for that question. So the model that I described earlier was on pro-social behavior in particular, but I think one of the most relevant issues is with antibiotic resistance. So in the Philippines, we all use antibiotics. I got a tooth extracted, I was taking antibiotics. If you eat meat, you're probably eating meat that was grown with antibiotics. If you raised a fighting chicken, it probably has antibiotics. And there's a lot of antibiotic use and misuse in the Philippines as well. Now, antibiotics come in different classes. Some of them are pro-social, some of them are not. Antibiotic resistance comes in different classes. So some resistance mechanisms are social, some of them are not social. Sometimes the bacteria secrete enzymes that also help the bacteria around them survive. Sometimes they just help themselves survive. There's no effect on the bacteria around them. Now, my research is on whether some of these kinds of antibiotic resistance are more favored when gene transfer is involved. So this antibiotic resistance crisis is a systemic problem. You can't just tell one person to stop using antibiotics. And systemic problems require systemic solutions. So we need to come up with policies, let's say, how bad does a problem have to be before we use this class of antibiotic over another? Should we be using different methods altogether? Should we stop using antibiotics on the chickens? Things like that. And you can only really start to quantify the pros and cons of this, if you have a good theoretical basis, if you have a good understanding of the dynamics of things like antibiotic resistance and gene transmission. This is not a hypothetical answer. I have an excellent undergraduate student, two excellent undergraduate students. One of them is over there right now. So she's currently studying, Miss Alex Sangalan is currently studying like are antibiotic resistance genes that are more prosocial, more commonly located on mobile genes, or are they not? And the other one on the screen earlier, Ms. Erin Echeverry, is doing it in culture, so she's trying to see if some antibiotic resistance genes are more favored depending on the kinds of mobile genetic elements, if they're more transmissible or not.
Can you wrap it?
Oh, okay, yeah, so does that answer your question?
Right, okay. The next slide.
Thank you very much. 3D printing is a very powerful tool because it allows us to rapid prototype a lot of objects. So in order to answer your question, my first answer is that the applications in sensing technology. Now, dual stereolithography, it prints very little structures. So it has something to do with compaction of materials used in sensors. This is very useful for the miniaturization of sensors. Now we can implant sensors in our body, etc., etc. And with the application of my chemistry, that would be more suitable to what is inside our body. Now, my second answer is that since 3D printing is now open more to masses, now this gives them knowledge to adjust, especially the properties of their 3D printers. Now, my third and last answer is that hopefully in the near future, when we talk about micro robots and nano robots, your robot will have three components. One, it will be the component to make it suitable for your body so that your immune system will not react against it. Second, it will have a detector in the form of MIPs that I showed you in order that it will attach itself to a receptor protein of your cell. And the third component is the drug in order to deliver the drug to wherever it is needed in the body. Thank you very much.
For me, we develop a material for oil spill remedy. I think the impact is very interesting. That we clean up the oil that has been spilled in our marine bodies. So first, we develop a biochar from waste material, which is banana peels. So instead of throwing it out in our garbage bin, so we can convert. We have the available technology to convert banana peel into a material that we can utilize for environmental remediation. And number two, that can help the whole of government approach when we are dealing with oil spill remediation. Because instead of sorting out adsorbent or material internationally, so why not we appreciate what we have here locally in the Philippines and use instead those biomass-related adsorbents because we have a lot of biomass here in the Philippines as an agricultural country, and through that simple tweak in their material properties, so we can directly impact the lives of our fisher folks and of course the people that are really affected by oil spill.
Sayang wala na si Secretary dito, no? Kasi pasok na pasok sa 6 P's ng DOST. So we have rare product, publication, patent, people services sector. Ano talaga yung 6 P's, no? But I think for an ordinary Filipino, the relevance, no, papasok sa saying na the Filipino family is one healthcare tragedy away from poverty. Remember, admission at the ICU costs 7,000 to 15,000 pesos a day. And to borrow an economic concept, the opportunity cost of that amount could have been used for other healthcare services and other priorities. No? And aside from that direct cost on the healthcare services, there's also indirect costs to the family. Bibisita ang nanay para bisitahin ang anak niya sa ICU. That costs how much? sasama si tatay, sino ngayon paano ngayon kikita ang pamilya? Those are the direct and indirect cause of this problem. That's why I decided to try to solve this problem. It's not gonna be a short route, definitely, but it's a step towards answering that burden.
Ang galing. Talagang palakpakan. Ate ko yun.
Dalawa sir, dalawa pag-usapan niya.
Okay.
Sige, go ahead, Dr. Alcantara.
Yes.
Okay.
Yeah, I'm Dr. Alcantara.
First of all,
So, to answer your question,
Sige, go
go
ahead.
ahead.
so to answer your question, as I presented a while ago, my research have already contributed to the development of the nasal spray formulation for COVID-19, which were utilized during the pandemic response in Thailand and supported efforts to enhance the frontline preventive strategies. So in the context of, in the Philippine context, this platform can be adopted not only for synthetic drugs but also for bioactive compounds, which are derived from our very rich biodiversity. So we have a wealth of medicinal plants which were proven to have pharmacologic properties. However, they have have a poor solubility and instability, and also the bioavailability, which often limits their therapeutic potential. So by applying these nanotechnology that I have presented, we can encapsulate these natural products to protect them from degradation and also to ensure targeted delivery. And moreover, this would create a pathway for commercialization of herbal-based nanomedicines, because as we all know, we have a lot of researches about herbal medicine, but none of them goes to the bedside of the patient. So this would offer a sustainable and homegrown solution to public health needs while we empower local industries. So it is not just about the innovation, but it is making the Filipino science work for Filipino health.
Thank you, Dr.
very
Alcantara.
much. By the way, I am very touched by your activity in Thailand. Yung service-oriented activities, hindi lang pinandapat nito yung pero ginagawa. I am really very touched and congratulations. May your track increase. Hindi po natin pagdating para. And then, Tiras man.
Hello, good afternoon.
Go ahead.
So the focus of my research would really be on the nutrient physiology of commercially important eukymatoids. So as we know, eukymatoids are the sources of sulfated polysaccharides, and these sulfated polysaccharides have important applications in several applications, for example, in pharmaceutical, food, and other applications. So also, this research would be on integrating these seaweeds in an IMTA systems or integrated multi-trophic aquaculture setup. So in an aquaculture, so there's an increase in nutrients that could have detrimental effects in our marine environment. So the focus of the research is really to integrate which strains or which species of seaweeds could be integrated in an IMTA system. Number one, to reduce the risk of eutrophication, and two, to increase the biomass for the increased biomass for seaweed farmers. Also, if the biomass is increased, then we would expect a increase in the revenues of our seaweed farmers.
Thank you, Mr. Alcantara.
Our winners. Let us give them a big hand of applause. Thank you very much.
Other questions from the judges?
Let me just ask some very short questions more to satisfy my curiosity. For Mr. Narvarte, in your study, because of your results, are you making recommendations on what type of seeds to more focus on? For example, Carbohydrates, Cruz had well, poorest performance, and maybe are you saying we should focus more on others?
Actually, sir, if based on our study alone, I think we would recommend the use of Capaphycus striatus because its ammonium uptake is not really affected by high phosphate levels. So in agriculture area, as I mentioned earlier, for example in Bolinao, Pangasinan, phosphate levels could reach up to as high as 4 micromolars. But for the two species, Capaphycus alvarezii and Euthuma dendrobilatum, their ammonium uptake actually declined at a higher phosphate levels. So, based on our study alone, we would recommend Capaphycus striatus because its ammonium uptake is unaffected. However, I want to emphasize that phosphate levels alone should not determine which species could be integrated in an IMPA system. Of course, we have to determine their growth, their tolerance to environmental stressors, their susceptibility to pests and diseases, and of course, their biochemical performance. We do not want to compromise those things. Sir.
Yeah, okay, thank you very much. Siguro si Dr. Parumino, if you're doing DZXL technology, do you have a setup here already in like in UST or some other?
Thank you for your question. To my knowledge, no, we still don't have a setup in the Philippines. However, in order to adapt, no, we can start with conventional stereolithographic setups, not one photon stereolithography. We have available setups, commercially available setups. For example, we can order from Amazon, and the resolution, it's satisfactory. It's satisfactory. For example, they can go as low as 10 microns. So anything that can be achieved within 10 microns. And then in the future, maybe we can develop a low-cost two-photon stereolithographic system. I showed the setup a while ago, so if we can have mirrors.
Do you have a time frame for setting up something like this in how much?
Maybe I would say within 5 to 10 years. That's just
Okay, iba ng technology.
yeah.
So that's how medyo behind.
Yes, sir. Well, another option that I could see is to team up with the university that I came from in France. So, well, I still have my professors there. So, yeah, we can establish a link. Yeah.
Maybe Dr. Lopez, because your question, do we have enough banana peels to clean an oil spill?
I would categorically say yes. Because in the Philippines, we are producing a lot of bananas. We know that, right? And in that banana, as I mentioned, so we throw 1/3 of that waste. And based on our result, a very small amount of banana peel biochar has a very high absorption capacity. So meaning to say, you only need a low amount of biochar so that I can capture those diesel oil that have been spilled in our marine ecosystem.
And maybe one last one now. Dr. Alcantara, in Thailand. The
Yes,
commercially available intranasal spray that was sold in development in Thailand, that uses the phytosan. Kasama yung Thailand.
Yes, yes, sir. It is included. Well, in fact, if you go to Thailand right now, you can see it in the supermarkets and even in local pharmacies. You can buy that nasal sprays.
So well, I suppose it's only for, I don't know, respiratory viruses, or you can also use intranasal for other types. Respiratory.
It is actually, yeah, right now it is actually used only for SARS-CoV-2, but we cannot use it for other influenzas or other viruses that are respiratory type. So it's quite specific for COVID-19 as of the moment.
During the COVID pandemic, in the Philippines, we would have this phrase, although we would hear some people recommend other types of sprays, the Catany or whatever. What's the reason why our medical community did not push for trying? Was there an attempt to create a nasal spray here for COVID in the Philippines? Are you familiar with that?
I think there is an attempt of using some essential oils. However, it did not materialize since I believe it is the lack of tools, infrastructures, and sustained support. Yeah. But if you are going to ask me, we can actually do it. We can just we just need to collaborate with industry partners or other researchers or clinicians. Yeah.
National scientists.
Thank you very much. I was very well informed and very happy about it. Let me ask Dr. Lincoln. Thank you very much. Dr. Lincoln, when you were talking about PGRMC2, you mentioned about what's present, the effect of PGRMC2 by cutting off the gene. And then, of course, you observe the result. But many times, the result is a collaboration between you take up the PGRMC gene. The rest of the genes will collaborate to supply and effect. So, therefore, what you did was not really causation. Although it is more interesting in correlation, but you could still have gene plasticity coming to your health. Is that correct?
Yes. So the question is, what prevents other genes from doing that certain function that is provided by
Also shall rule here, so that the others could come together and help supply the results of the gene that was suppressed, as it were.
Yes, actually that's correct. So we didn't show it here in this presentation, but of course we look into progesterone receptors. Again, fetal embryos don't have the classical progesterone receptors, but of course, if there's PGRMC2, what would prevent PGRMC1 from rescuing that function that is caused by knocking down or deleting PGRMC2? So in our laboratory, it's one of the results in the appendix. We actually did PGRMC1 deletion, as well. And we should in here, even when we did PGRMC1, we not see any of these effects. So it is only specific to PGRMC2. Now when we caught both PGRMC1 and PGRMC2, the effects that we were able to see persisted. So while I agree that it probably isn't just the fault of one molecule, this molecule definitely contributes to that event leading to the activation of the pore.
So you're saying that the manifestation of PGRMC2, you actually looked at PGRMC1 and worked on it, and in fact did not see any results. But it could be that multifactoriality expresses itself in PGRMC3 or 4 or 5. Unless you have control over this.
So when, of course, there's going to be the cascade progesterone receptors, and there are only two complexes. So 1 and 2. So if we remove one, it's only definitely the 2 that's going to be binding to that progesterone. So we activated what we see because we technically what the CRISPR-Cas9 provided is a loss of function. And that's how we do functional genomics. We or modifying that certain gene to see what are its effects, because when we needed that control for all other conditions, that might undo its function. Definitely more studies are needed to provide more specific pathways that are involved in this. This is actually one of the future directions that I'm going to take in establishing my lab here in the Philippines. So we're definitely going there, but those are the unanswered questions that we still have at this point.
You still have to look into it. The other is bacteria. I'm flabbergasted that bacteria cooperate. It's modern biology very strange stuff. Because you have Michael Levin saying that all our systems are actually collaborative systems. I'm a trained communist, but I no longer consider myself a communist because I have, in a sense, divorced from economics as the foundation of economics. I'm already moving towards sociality. I don't work on computer simulation and invent methods. I only work on theory. In theory, pro-sociality emerges in the population when selfishness becomes punished. So when the system starts to punish selfishness in the population, but without punishment at all, selfishness will come. Like your yellows will dominate the population. What is the equivalent of recombination in theory? Is it punishment? Yes. Or maybe good recombination, right? Good and bad.
It's a bit less tied to punishment and more tied in with the idea of homogeneity. So it's a homogenizing force. And effectively what happens is if you have like a lot of selfishness in the population, they kind of punish themselves, right? They don't reap the benefits of cooperation. Whereas
I don't understand the meaning of punish themselves.
let's say you have like two groups. This is group selection. If you have one group that's all selfish, then they have lower fitness than a group that's all cooperative.
Only cooperators will have lower fitness in that group.
Yes, so between groups, cooperation is good. But within groups, cooperation is less faithful, right? And usually to maintain cooperation, the classical explanation for it is like you have to punish the selfish actors. But in this case, you don't need to punish them because you can change their behavior by changing their genetic material. You change what they are by definition. If there are enough cooperators and these cooperators have this recombinant ability, then in like if they change the cells near them to also become cooperators instead of selfishness, then they gain a relative advantage over other groups that don't have this capacity to change the cell.
Trigger for recombination.
Sorry,
Trigger for recombination.
In nature, some bacteria is produced by default. Sometimes it can be in response to stressors. So it's just a way for bacteria to collect genetic diversity. So sometimes in case of they feel like they're dying, that they might engage in more, they might start taking off more DNA from the environment. And for the conditions that you described, I think like the selfishness might act as like a pressure for them to recombine some more under some conditions.
In theory, if you do not punish selfish behavior, you're same as the iron law of selfishness. In other words, the whole population will be overrun by the selfish genes, as it were. So you have to have some pushback against selfish behavior. And I was trying to understand what the combination is it analogous to punishment or selfish behavior?
It's not the same as punishment in that you don't impose a cost onto the selfish cells. You just pressure them into not being selfish. So for the human systems, I think it's more similar to like peer pressure. In which case it's not like direct punishment, right? It's like if you see everyone around you behaving pro-socially, then you don't like feel kind of ashamed or your selfish behaviors that's likely to come out. Except in this case, the mechanism isn't behavioral or social, it's genetic.
Okay, so there's a difference in implementation. So combination is not punishment.
It is not
So I will beg to disagree. Because in theory, if you don't punish, you will be open. Because the population of pure opiates will be invaded by mutants. One mutant in a population of opiates will become very rich, will become very thrive. And therefore its posterity will increase. I don't know how we will
Can I request? So we showed earlier, like in the image, that we do show that in the absence of recombination, the selfish cell can increase in number and it will come to dominate the population. So what happens here is there's a trade-off between the relative fitness benefits you get from being selfish, so classically, and this acts against the homogenizing force of recombination. So it does have an advantage, but this advantage might not be enough to overcome the genetic peer pressure from all the cells around it. If it doesn't reproduce at a rate that exceeds the rate at which it's converted back into cooperating behavior, then it becomes a cooperator. If it exceeds that, then you see the what's normally expected, in which case it takes over the population. I'd like to clarify that the combination can help promote cooperation, but it's neither necessary nor sufficient for cooperation. If the other conditions don't allow it, then it just won't happen.
Anyway, so, because I thought you.
Yes, I'm sure you have any
Couple of short targeted questions. The first one is for Dr. Lee, since we're talking about your work. Do you think that problem can be simplified enough that you could solve it analytically using a theory without losing the fidelity to real active behavior?
The field of evolutionary game theory came up because, like, it started developing because the biologists, when they started using analytical game theory to describe biological systems, found that, like, the bacteria aren't rational, so it doesn't really work that way. So, some things that would fail to invade the population because they are rational actors might work for bacteria because, again, they're not rational. Their behavior is genetically encoded, which is randomly determined at first, and then the payoffs are settled later. They don't calculate the payoffs beforehand, right? So, the analytical game theory serves as a framework, and lots of the expectations are met when you're talking about, like, in biology and evolutionary game theory. But I believe the current consensus is that it's not sufficient to explain the behavior of systems like that.
Thank you. I'll move on to my next question, which is for Dr. Lopez. I'm interested in the genesis of the idea that you presented. Why banana peels and not some other biomass resource?
The number one reason why we chose banana peels is because of the abundance in the field. So because in order for us to make an impact locally, so we need to choose a biomass that can be converted to biochar as a natural. That should be abundant. That answered also the question earlier, whether we have a sufficient supply of that raw material. Because there are other biomass that are available, like the lamb, sugar cane, and coconut. But if you're going to transform that into a biochar, then have a really an impact to our society. So our raw material should be stable enough so that if ever there will be an oil spill that happens, we can easily deploy that to the site of contamination. And number two, is because of the properties of banana peel. So we were able to characterize banana peel. So it is very rich in functional groups as well as very high carbon content that is suitable for conversion to bio.
Thank you, Lemicio. Any questions from the judges?
So much to learn from a foundation where I asked a question it's nice that we saw this in different groups because if you were to do a biochar, then it's a way this will also chemical transformations there, and the surface must be rich in functional molecules. But I'll just ask you a question. Will be absorbed diesel oil? Will be absorbed? What will happen then with the banana peel biochar? Will the diesel oil that is being absorbed? So you say it's the radiation and I suppose it's the acceptance. What will we do with that?
That is a very good question. Actually, once we have the banana peel biochar, once it was able to absorb the diesel oil from contaminated marine site, for example, so we can actually collect biochar. And then from that biochar, the oil is only absorbed on the surface. So we can actually do some treatment or recover that. And then once the oil is removed from the biochar, so we can reuse it later on. So So that's the beauty with adsorption because this is a chemical process. So unlike in a pure chemical process where in the chemical, so here we can recover the diesel oil that was spilled from the environment. So we can reuse it. But for our studies, so we haven't delved deeper into that, but we can do it in the future for regeneration.
Sandali lang. Are there any other players aside from these two fishes in that might contribute? The second question is, what's the most common trigger of premature labor? Is it really lipopolysaccharide, or is it smoke, or is it infection in other conditions? So, is your model good?
Thank you for the question. So, with regards to the first question, about Doctor, what's the first question?
Other tissues, aside from those two major ones that you say are talking to each other, interacting. So, are there others? Like, because labor is about muscular contraction.
Yes, absolutely. So,
where are the muscles? And where's the breathing? Because, labor is really something that you experience pain with. So there must be nerves.
yes. So we chose this specific tissue because once there is contraction, it's already too late. You are already at the effector side of things. So we have to address it before it goes to the increase. And I chose this specific tissue because one, it's understanding no one has really studied this extensively. But in order to also show that all possible these tissues can contribute to premature activation of labor, we also developed other organ on chip in the reproductive system. So we already have a placenta on chip, a cervix on chip, and a vagina on chip. So we have all those models to be able to see the contributions of all of those organs to premature activation of labor.
Okay. Now, you're monitoring gene expression. So it's just RNA, right? So is there part of the paper that also gets the levels of the actual hormones or
Yes, so aside from the expression of aside from the gene expression, which is only at the level of RNA, of course, there's going to be proteins in there. So we also measured one, the level of hormones, especially progesterone, which is important in pregnancy, and the level of inflammatory and anti-inflammatory molecules, such as cytokines, soluble in change.
Thank you. So I wanted to ask Isaiah, so the model of bacteria that we use, are they all exactly the same? They're from the same species, they have no different strains, they have exactly the same genetics. So why would they combine? I mean, I've been studying bacteria myself and I know about the bioinformation. And these are prokaryotes trying to be multicellular animals to defend themselves. Secondly, the principle actually of the functional technology is balancing cooperation with competition, because there must be a scenario where the best would survive, and that's the mutant. And then that's the one with the genetic mutations from whatever sources, not necessarily from recombination, from natural horizontal transfer. So many things are triggered by the environment. Third is, of course, symbiosis. The true progress in evolution has to do, Professor National Scientist Peru, with a cooperation form of symbiosis of two different types of organisms. And some of the bacteria actually lose their genes and they become choosing rivals. I didn't know that. So maybe quick answer.
Okay, that's a lot of questions. First of all, for the model, they behave exactly the same way except for the combination behavior. But for like our current lab experiments, our RA managing those here at the moment. Say hello, Ray. Okay, so here we're working on E. coli. We have like strains that are more prone to recombination and the strain that's less prone to recombination. But they're all E. coli, and we use E. coli because it's easy to work with, and we sort of have like tunable levels of gene transfer. So if you wanted to transfer more, or if you wanted to transfer less, that's pretty doable in the lab for sure. In the lab, yeah, in the lab. Your second question was about sort of like the balance between cooperation and competition, right? In this case, the interesting thing about gene transfer is that it's both rolled into one. You transfer a cooperating gene, and at the same time, because you change the genotype of the other organism, you are removing the competition and replacing it with essentially a copy of, it's not a full copy of yourself, but it's like related to yourself genetically because it has a portion of your genetic material already. So it's both. There's
Two levels of cooperation because when we find a biofilm or release small molecules, there are messengers or basis of interaction. That's also genetic down the line. I mean, it's from the transcription factor, there's a series of genes that are coding, genes coding a small molecule, and that's quick. That's very quick response. Anyway, it's just to let me think about it.
For the third question, I think this is it's kind of related in that the symbiont and the host will sometimes exchange genes one goes to the other, and this is kind of like I think it's the esteemed academician mentioned earlier that it's like punishment is required for cooperation, and this is I actually think this is punishment of a different kind.
Punishment is just feedback.
In that
punishment.
it's
Try to interpret it as punishment.
okay. This case of gene switching creates an interdependence in that if one of them tries to betray the other, it can't because it's going to get punished because its genes are no longer in its own genome. So it's a threat of punishment.
There's a lot of violence alone where complex form pathways are full of reward and punishment, positive and negative feedback, right? So there's the violence as well. Anyway, thank you. So, other mathematician solved the problem and probably get what is that? No, the prize. Yes. So, anyway, it's just reading about these elliptical curves and they have distortion points. Are you aware of that? That's false field of study also. Anyway, you saw understand the distortion points in the elliptical curve using your small monster or your big monster. So you make the mean, algebra and then geometry.
Right. So torsion points. So elliptic curves, they are not just curves, but they are also groups. You can perform addition on these points. So there's a way to add 2 points and get another point. When you say torsion point, that means that if you add to the point itself enough times, we get back to identity element. So there are algorithms to compute these torsion points that don't rely on this connection with the monster group. But I have not really thought if there is a way to do that using the algebra or the properties of the monster group. because when I have to compute them, then I can do so without having to rely on external.
Thank you. I have to ask Dr. Alvaro. Dr. Alvaro,
Yes, ma'am.
congratulations. Tells you the scientific and technological capability issue along our university. None of those experiments are still being done in any university in the Philippines. So congratulations. Yes, and we are able to transfer technology here and very good. But I just wanted to know why it's FVR, that is user model antiviral drug, and actually by itself, it's in other studies not shown to be a good COVID drug. And I know it's depends a lot on the mode of administration. And in this case, we really just want to deliver the drug in other form. Now, I didn't read your paper in great detail, there's really no time. But the idea of using prednisone and alginate for one, is that the case of slow release of the drug so that we do not have this kind of toxicity that we talked about when we introduce the nose and the respiratory tract? Would you say that there should be...
Okay, so the first question.
Be the appropriateness of the antiviral drug. The second is the appropriateness of the function of the nanoparticle.
Okay. Okay, so first of all, the appropriateness of public review. So let me tell you that this research was during the height of the pandemic, the early 2020. So we don't know much about the virus. And the FDA during that time, the Food and Drug Administration, only allows repurposing of antivirals. And one of those antivirals that were used during the start of the pandemic was Favipiravir. Hence, we focus on this one. However, the one that was commercialized is already an antibody against COVID-19. So it is more specific to the target antigen, which is the SARS-CoV-2. With regard to toxicity, since we use a non-toxic and natural polymers to encapsulate these drugs, the favipiravir, so we have seen no toxicity to the nose, and yes, that was correct. That is due to the slow release of favipiravir in the noses.
Okay, thank you.
And what's the last question, ma'am? I'm sorry.
I think there are only two questions.
Okay. Thank you.
Dr.
Thank you, ma'am.
Dr. Lamarque from the Philippine Marine Science Institute. I saw you flash some kinetic data. KM or K. So can you please explain that better? What are you measuring? Which is your biochemical catalyst here? Please explain that, because the table is not in your, it's not here.
It's not in the paper.
It's in the supplementary material? Is it?
Yes, ma'am.
Just please explain.
I use that data to support our explanation because in the paper, we found out that Epiphytus striatus had the lowest range of phosphate uptake, and we attributed that to the low surface area to volume ratio of our samples. However, in this study, we were not able to directly measure the surface area and the volume of the samples, but in another study, in which we were able to have an estimate of the surface area and volume of the samples, we determined that strains with high surface area to volume ratio had faster uptake rates. Also, they also exhibited lower KM values. The samples with low surface area, sorry, high. Samples with high surface area to volume ratio, we found out that they have lower KM values, indicating their deficiencies in nitrate output.
Alright, just want to ask if you're familiar with the Roman or Kawikawi seaweed farmers. They are in dire need of help, and I have already communicated with your mentor, Dr. Roletta. Okay, so what are the species of Ukima found in Kawikawi?
As far as I know, the most cultivated species there are Copaphycus albarazii, Copaphycus striatus, Eucuma, and also Copaphycus malicianus. What's interesting is that previous reports would tell us that Copaphycus malicianus is only found in that area of Philippines. However, our recent studies suggest that it is more widely distributed, and I think this would have an implication on the strain selections of our crops, no? Which strains would be would survive or would be good for cultivation, mass cultivation.
So please keep the call, okay? Communicated to UPs about livestock farmers. Thank you. Dr.
Thank you, sir.
So I've had some experience working with HDMA, hydroxy it's an acrylamide. These are small drug delivery carrier systems. They're not capsules. And the great thing about that is that we can get them to enter or do pinocytosis instead of endocytosis. The property of that small drug delivery system functionalize, so you can actually attach it on the first one. So in your case, your acrylic and acrylic structures, you don't have only the alkene group developed on. So would you consider for broader applications using other materials like the hydroxy version?
Thank you very much for your question. In fact, in my paper, I used acrylates and pentacrylates because at the time those were the most common photolysis for PPS. But the good news is that for acrylamide, I see that this is very possible. I haven't done it because it also has the double bond, C double bond, and it's conjugated to an electron withdrawing group. So in the case of acrylated metacrylates, the withdrawing group is the carboxylic. This one is the CN group. So I expect the chemistry to be the same. And the photoinverter that I use will be usable on each PPS.
So what's the first product that you're working on? Because you talk about so many potential applications. So what is your first product?
Now that I'm back here in the Philippines, I have 2 students in the coming academic year. We're planning to work on photonic crystals. So these photonic crystals, it's like what I told you a while ago, compact-based CDs. No? So they change color depending on the angle, but not only that, they change color if the microstructures within them change. And it's possible to induce change in the microstructures if it recognizes a target molecule. This will lead to the change in the structure at that level. Thank you very much.
Dr. Naconis, so both of the questions are very serious ones. And are they also very susceptible to lateral or horizontal gene transfer? Do they mutate as much as bacteria? And the second question is, what would be the crop that you would like to work on, for example, banana or some other major crops, for local consumption and also for export?
Thank you for your conception. So in the fungal, if you compare to bacteria, they relatively have a bigger genome. And so, I mean, I guess I would expect that for the genome of bacteria, it's about maybe like 10 million base pairs. But for fungi, an average would be 40 million base pairs. So they're kind of huge. And so the idea of horizontal gene transfer may still not be the consensus, I guess, for every fungal plant, particularly for fungal plant pathogen. But what we've seen in Fusarium oxysporum, which is a known fungal plant pathogen across many different species, not just in the plants, but also in animals and even in humans. They have this what we call as similar to bacteria, like a pathogenicity island, and we refer to this as conditional dispensable chromosomes that carry pathogenicity. In a study that is being done by the group at I think University of Michigan, they were able to identify this particular region that's extra chromosomal, where when they have swapped this different, or like when they swapped this pathogenicity island, they were able to identify that they are capable of switching hosts. So indicating that, because for example, if you say Roxas it's a species complex. And in this species complex, they are varied based on the host of where they can infect. And swapping this in this dispensable chromosomes can actually alter their host species. So an indication of maybe it's possible for horizontal gene transfer, but it's not similar to the idea of like plasmids that can just easily shoot up, similar to bacteria. Now, with the second question, I've actually been working on not specific fungal plant pathogens, but those that are causing emerging epidemics. And so, in particular, this with the Neopestalotiopsis that I mentioned earlier, it's very similar with Corynespora crassicle, which I mentioned in the study, because they are asexual fungi, and yet we are seeing these phenotypic responses in terms of their variation in how they respond to a certain host, to a certain fungicide, for example, and yet they're clonal. So what's giving them the ability to respond differently if they're asexually reproducing? So currently I'm working with the Neopestalotiopsis that's affecting the rubber and the strawberry. And then just recently, I have been working with people from the University of Southern Mindanao to work on the Cigatoka pathogen. So this one in particular is affecting the banana industry, where fungicides or resistant to fungicides are a main issue, especially because while in some crops, it's an option to spray fungicide. But for bananas, especially those that we export, Cavendish for example, it's a requirement. And they are on the basis of calendar spraying, just because you don't get any yield if you don't spray.
So to say, there's a lot of scientists here must have this mission to share their knowledge, not just with their universities or their research groups. I mean, we're on a step up mode. We are important to know in our country, so we must really go out and look for universities, other students outside your universities. Thank you very much.
So, national scientist for very last question. Short question, short answer.
Dr. Lee. No, sorry. Yeah. The monster. You alluded to baby monster. Presumably there are adults, mature monsters. And there are different sizes of this monster. As I don't really get what is being counted here. Are you counting the symmetries, or are you counting much less CERN, the triples does not exist for n greater than 3. 3 or better. But for n equal to 2 or 1, it's infinite. Has it anything to do with this?
So what's being counted here is the number of symmetries.
Number 2.
Right. So for example, in the triangle case, we had 6 symmetries. So that's the order of the corresponding group. In the case of the monster, there's this algebraic object that lives in 196,883 dimensions, called the moonshine module. And the number of symmetries of that thing is the order of the monster. That's what it's on.
Okay. Thank you. It was a wonderful afternoon.
Okay, so that concludes our.
I think you will have something to say.
Yeah. So that concludes our open forum, and we thank our board of judges for asking wonderful questions that really highlight the scientific contribution of our finalists. So at this point, may we call on kindly